Hi, this is Marley Romero for Microbiology. Today is March 22nd and it's 12:51, and I'm going to be doing the staining technique. And for this first experiment, I already wrote on my slides, as you can see, and I drew my circle. Sorry, I felt all jittery, nervous trying to write on it. So what I'm supposed to do is grab a sterile Q-tip and then scrape the inside of my cheek, gums, and teeth. And then what I'm gonna do is on my slide, and the circular motion, I have to rub the Q-tip on here inside the circle to make sure I'm getting all the cells on here. And then I can set that down or throw it away. I have to fix my slide, so carefully I have to pass my slide with my little clamp so I don't get burned 5 times. 1, 2, 3, 4, and 5. And I have to set this in my little area, let it cool for at least 10 minutes before I can actually put my stain on it. So I'm gonna pause the video and then record once I do that step. While I wait for my other slide to cool down, because I don't want to break the glass or do anything crazy like that, I grabbed a new Q-tip, sterile Q-tip, and I did this again to collect a sample, rubbing your gums and teeth. And I'm gonna smear my sample here, and I'm gonna add 1 drop of the Congo red. And I'm gonna agitate it by kind of rolling it around to make sure that it goes on my sample. I think this is how you're supposed to do it. I kept reading and reading the instructions, but I was a little confused on this part. Then I'm gonna grab my clean slide and grab that drop and then slide it down. And then lace this on, and I'm gonna just let it air dry. Don't mess with it. Once it's completely air dry, and I should see it like dried up inside, then I can look at it under the microscope so I can see what the cells look like. And I'm gonna go ahead, and the 10 minutes are up, so I'm gonna go ahead and move on to staining my first slide. Okay, so now that it's been the 10 minutes, I placed my slide in my little tray using the crystal violet gram stain dropper that came in my kit. I'm gonna put 1 to 2 drops stain inside the circle. I'm doing 1 just because I don't want to mess it up, and it looks like it's kind of spreading out on my circle, if you can see. And I'm gonna just let it sit here for 60 seconds so that whatever bacteria or cells that are there kind of absorb the stain. And then after that, after the 60 seconds, I'm gonna go ahead and hold it up at an angle, and I have to rinse off the stain with water until the water runs clear, which should be okay with 1 pour of water. So I have my water in my cup here, and then I can go ahead and look underneath the microscope. So I'm just waiting. All right. So now I'm gonna put my slide at an angle. So like this. And I'm gonna pour the water over so it runs off. And it said you can blot off the excess water, so carefully I'm just gonna touch it like this. I'm trying not to touch anything on or near the circle, and underneath I got water on there, so I'm just kind of drying it off. And already I can see tiny little purple dots in there. I don't know if you can kind of see. You can already see purple dots. And I can go ahead and look underneath my microscope. Careful that I do not let go of the clamp holder hard, because it can break my slide. Well, I can't see with my goggles. Oh yeah, I can see. It is coccus shape bacteria. I'm seeing coccus shape, and this is on the high power. And if I switch to the low power, I'm kind of blind, so I can't really see nothing. I still can't really see much, except for only on the high power. So I can then take my photo as needed for my lab assignment, and that's it for my staining technique. And that's a wrap.